welcome to the discussion class of biochemistry. Next we'll be talking about the configuration and conformation. We need to understand these two terms to understand a process of denaturation. We had seen in amino acids that amino acid can exist in two forms, D form and L form. While we had been talking about these D and L form, we saw something about this chiral atom, the chiral carbon. Chiral, that means asymmetric, and this asymmetric carbon had various side chains attached to it. So it had all different side chains attached to it. Because of its chirality, it could exist in two forms, that is D form and L form. Now very important thing about this is whenever a molecule, be it amino acid or thing, whenever it has to exist in D form and L form, it has to change between D to L or L to D. There has to be breaking of covalent. It cannot just turn around to from D and L. It has to break its covalent bond. This is called as configurational changes. But we have one more term called as conformation. In conformation, what happens is this is basically about spatial relationship of every atom in a molecule. We take a rather simple example rather than amino acid. If you take an example of water molecule, this is a water molecule, that is H2O. But how does it exist? It exists in a typical conformation. There is a typical angle between these oxygen atom and hydrogen atom. It's because of their size, because of their charges and their attraction, polarity, and all those factors. They exist in a typical structural shape. This is called as conformation, and this conformation can be changed without breaking the bonds, and it can be changed just because of the rotation along the single bonds. So in denaturation, what happens is there is no change in the configuration, there is no breaking of any bonds, there is only loss of the conformation of the protein. And this conformation loss is because of the rotation along the single bond, and that results in denaturation of protein, causing loss of activity. So what we saw was change in configuration needs breaking bonds, change in conformation needs rotation around the single bonds. It does not need any breaking of bond. And in denaturation, what happens is change in conformation of the proteins, resulting in loss of activity. With this, we go ahead to study some of the important proteins. We'll be talking about the chaperones, the hemoglobin, the myoglobins, and the cytochromes in today's discussion. So chaperones, what are they? They are the proteins. They are the proteins that help another protein in non-covalent folding or unfolding. What does that mean? We had seen the primary structure of the protein, then we had seen it forming the helices and the beta sheets. When it was forming helices and beta sheets, there was hydrogen bond that helped in folding of the alpha and the beta sheets. But this hydrogen bond, very typically, it got incorporated in the protein itself. But in chaperones, what happens is they are the proteins that help from external, that help from outside in folding and unfolding. They do not themselves get incorporated in the protein to form the part of the protein. They just act from outside. So you can say these are the chaperones. They themselves are some kind of proteins, and they just help from outside in folding and unfolding. But one more very important thing about is when a particular protein, for example, you have this protein is performing its function. When it is performing its function, chaperones will do nothing. But if something happens because of which this protein gets stressed, chaperones, they come into role and they try to protect this protein and prevents its folding or unfolding and prevent it to become useless, prevent it from becoming useless. And this typical role of chaperones, they give them a name. And all this happens especially when the native protein is stressed. What kind of stress it could be? It could be a stress in the form of heat, it could be stress in the form of pH. And whenever these stressful conditions exist, chaperones come and help them, prevent them from becoming non-functional. Because of this, chaperones are many a times called as heat shock proteins. And these heat shock proteins or chaperones, they are expressed by the cells. They are expressed by the cells when they are under stress. These heat shock proteins, they are formed by the bacterial cells. They are also formed by some of the human cells, and they are mostly formed when the cell is under stress, because they are important for preventing its other functional protein from becoming non-functional. So the major function of chaperones is to prevent the newly synthesized polypeptide chain and the assembled chains from forming the non-functional structures. And this tendency is typically increases when there is stress. So they are chaperones, their expression is increased